Welcome back to Chapter 9, Part 1. In this lecture, we'll look at microbial growth. This chapter covers how microbes grow, as well as their requirements for growth, such as pH, oxygen, temperature, as well as other requirements. We're also going to look at the type of media required for growth. But in this video, we're going to focus on 9.1, microbial growth. Chapter 9 covers binary fission, growth curves, generation time, methods of measuring microbial growth, special cases like budding and fragmentation, and we'll finish with biofilms and quorum sensing. This material sets the foundation for understanding how microbes multiply, survive, and sometimes cause disease. Bacteria reproduce asexually through binary fission. Before dividing, the cell replicates its DNA, starting at a region known as the origin of replication, and that's indicated in red in these chromosome pictures here. As the cell elongates, gets longer, these duplicated chromosomes are pulled to opposite ends of the cell. And a protein called FTZ, or FTSZ, is going to assemble into a Z ring at the middle of the cells, and this will direct where the cell wall and septum will form. This Z ring is essential for cytokinesis, division of the cytoplasm. At the end of binary fission, the parental cells splits into two genetically identical daughter cells, and this process is fast and efficient, especially in favorable conditions. You'll notice sometimes the cells don't end up separating. They might stay together, and this is where we get arrangements such as pairs or chains. Here is a closer look at binary fission, and I can see the origin of replication a little bit more closely here, here in the chromosomes. Here are the FTZ proteins, and they're going to assemble to form that Z ring that guides the formation where the location of the new septum and cell wall will form. Eventually, the cells will separate and form the two daughter cells. So the name of that protein is the FTZ protein, FTSZ protein, that forms the Z ring to direct septum formation and cell division. So that septum in binary fission is that new dividing wall that forms between the two future daughter cells. The Z ring guides the inward growth of the cell wall and the membrane and creates a partition, a separation or septum that gradually constricts and separates the two cytoplasms. The generation time or doubling time is how long it takes a population to double through one round of binary fission, and this varies widely. So E. coli, for instance, can double in about 20 minutes at optimal conditions, whereas Mycobacterium tuberculosis can take up to 15 to 20 hours to double, and Mycobacterium leprae can take 14 days. These two generally take longer because of their mycolic acid walls. We can calculate the number of cells ultimately formed using this equation here, where the final number of cells formed is equal to the initial number of cells times 2 to the nth power. And the small n over here, that's part of 2 to the nth power, stands for the number of generations that have occurred. Let's look at that equation a little bit more closely, where we'll just look at 2 to the nth power first. So if I start with just one cell, remember the final number of cells is equal to the initial number times 2 to the nth power. If I only start with one cell and I have no division, let's say n equals 0, any number to the 0th power equals 1. So I would just have one cell if there's no division yet. If I have one cell and it divides once, that would equal 2. So after one generation, one round of division, then I have 2 cells. And if I have one cell to start with, but it divides twice, I would get 4 cells, and I see that here, and so forth. So let's try this check your understanding question. If we have a doubling time of 30 minutes and a starting population of 1 times 10 to the 5th cells, so that would go in here, how many cells will be present after 2 hours, assuming no cell death? Please try that on your own, and in the next slide, we'll go over the answer. So let's solve this together. I hope you had a chance to pause that video and try it on your own first. And I'm looking at doubling time. That's my generation time, 30 minutes. and I'm looking at how many cells I have after 2 hours. So the first thing I did was convert 2 hours to minutes, and that's equivalent to 120 minutes. I know there are 4 generations that have occurred during this time because it takes 30 minutes for the cell to divide, and 120 divided by 30 is 4 generations. 4 binary fission cycles occurred. If I plug my numbers into this formula, the original number of cells I said was 1 times 10 to the 5th, and then 2 to the nth power, I had 4 generations here. So 2 to the 4th power is 16. I'm going to multiply that by the original number of cells. I'm going to get over 1 million cells. So 1.6 times 10 to the 6th power cells. That means I went from, this is 1 times 10 to the 5th, which is 100,000 cells, to 1,600,000 cells after 2 hours. When you grow microorganisms in a closed culture, so closed culture, like a test tube or a flask, in which you don't add any new nutrients and you don't remove any of the wastes, you're going to see a reproducible growth pattern, and we call that a growth curve. There are generally 4 phases that you're going to see, including the lag, log, stationary, and death phases. In the lag phase, you don't see any significant increase in the number of cells. They're kind of getting used to the new environment. In the log phase, you'll see an exponential increase in the number of cells. Eventually, they're going to reach a plateau known as the stationary phase, where the number of new cells is about equal to the number of cells that are dying. Finally, we'll run out of nutrients, wastes will accumulate, and we'll see the death or decline phase, where the number of dying cells surpasses the number of dividing cells. All right, let's look at these phases a little bit more closely. Remember that when we plot the number of living cells over time in a closed culture, we're going to see 4 distinct phases. The first phase being the lag phase, where cells are metabolically active, making enzymes and repairing damage, but they're not dividing yet. Sometimes you'll see a slight increase in the slope, but very insignificant. Then we have our log or exponential phase, where the cells are dividing very rapidly by binary fission. Growth rate is maximal and uniform. This is also when cells are most sensitive to antibiotics. During the log or exponential phase of growth, the relationship between time and the number of cells is not linear, but exponential. You can see the steep increase in growth. But the arithmetic or linear y-axis produces this steep curved line that makes it hard to analyze. So instead, we often plot microbial growth on a semi-log scale. Here's my semi-log scale. Time is normal. It increases by increments of 1, whereas the y-axis increases logarithmically. So every set of values, is 10 times bigger than the previous set of numbers. This makes trends and calculations much easier to see. So the numbers of cells will increase by constant multiples during exponential growth, and our data will become a straight line, letting us really read off the growth rates and doubling times more directly, compare different cultures more easily, and see both small and very large cell numbers on the same graph without having to compress the data. In the next phase, we have the stationary phase. This is when nutrients start running low, waste accumulates, and the rate of cell division equals the rate of cell death. Cells might produce endospores and virulence factors at this time, so these cells are currently in survival mode. Finally, we have the death or decline phase. This is when toxic wastes accumulate and dying cells will start outnumbering dividing cells. A few persisters sometimes will survive, and these are microbes with slow metabolic rates. Both in the lab and industrially, we generally want to keep cells alive and in the log phase of growth, so we use devices like chemostats, and chemostats continually add fresh nutrients and remove wastes and dead cells to sustain exponential growth. So we saw the phase that growth occurs at the fastest rate was the log phase or exponential phase. And then the second one, 2 factors that can limit microbial growth. One could be nutrient depletion. We ran out of nutrients, so the cells can't divide. Or another could be waste accumulation, right? Toxic waste accumulation. Sometimes oxygen limitations can also contribute to limiting growth. So we can directly measure how many cells we have by counting cells. And we can directly count cells using a Petroff-Hauser chamber, which is like a hemocytometer for bacteria. We'll place a measured volume of cells under a cover slip and count cells in grid squares to estimate their cell density. But because not all counted cells are alive and you can't really tell the difference between a living and a dead cell under the microscope, we sometimes use fluorescent stains, where living cells will fluoresce green and dead cells will fluoresce red. We can also use a Coulter counter, and this is an electronic device that counts cells by detecting changes in their electrical resistance as they pass through a tiny opening. Overall, these methods are pretty good at estimating total cell number, but it's really difficult to tell if these cells are living or if they're dead cells. Here's an example of that Petroff-Hauser chamber. If you were to zoom in on either of these metal plates, you would see grids that appear like this. Once you put your sample on there, you would count the number of cells in different grids. Here they're showing you one particular square and there are 10 cells in there. You usually will count multiple grids, so the number of cells in more than one square, in order to get a pretty reliable average and reduce the error due to uneven cell distribution. But this doesn't always yield an accurate count of the number of cells because you can't really tell between living cells versus dead cells or even debris if they're the same size as the cell. Fluorescent staining can help us overcome that barrier and help us differentiate between living cells and dead cells. The primary stain is the green one, and this can penetrate intact cell membranes, so both living and dead cells can stain green. But the secondary stain in this process is going to be a red fluorescent stain, and the red stain only stains a cell if the cell membrane is very damaged. So live cells will end up fluorescing green because they only absorb that green stain, whereas dead cells will appear red because the red will displace the green stain when they penetrate those dead cells. And here's an example of a Coulter counter. It allows cells to pass through the small opening and measures the resistance change as the cells pass through. But again, this one can't really tell the difference between living or dead cells. So any cell that passes through, whether live or dead, will show a change in resistance. All right, in this checkpoint, we're going to look at why we count more than one square in that Petroff-Hauser chamber, the one of those grids, to estimate cell numbers. The reason is so we can get a reliable average and reduce error that might occur due to uneven cell distribution. If there happens to be too many cells in one grid and too few in the other, this will average out if we count multiple grids. In the second one, that fluorescent staining, why do dead cells appear red? This is because the red fluorescent stain only penetrates the damaged cell membranes, the cells with those damaged membranes, so it'll displace the green stain on those cells. So live cells appear green and dead cells appear red in the staining method. To count only live cells, we can use viable plate counts. And these are expressed by counting colony-forming units, also known as CFUs, per milliliter. We do serial dilutions, and then either the pour plate method or the spread plate method. And I'll show you these in a bit, but the pour plate method is when you mix the sample with warm agar before it solidifies, and colonies will form both inside and on the surface of the agar. In the spread plate method, we spread the diluted sample across solid agar, and colonies will form on the surface. And then we'll count the number of colonies on each plate. We aim to count a plate with between 30 to 300 colonies for statistical accuracy. For very dilute samples where you probably won't find very many microbes at all, like drinking water, we use a process known as membrane filtration, and this is going to concentrate microorganisms on a filter before we place it on agar for growth. So in this picture, I have a serial dilution shown here. Here's my original stock, my sample, And then here I have tubes with just media, and it looks like 9 milliliters of media per tube. Imagine it's sterile media at this point. I'm going to take 1 milliliter of my stock, my original sample, and put it into 9 mils, making a total of 10, a volume total of 10 milliliters. So that's a 1 in 10 dilution. And then they do the same thing. Now they're going to mix this up and down several times and pipette out 1 mil and put it into 9 mils of the next solution. And if you look closely, this is going to happen over and over again until we get to the last tube. And the reason that this tube is lighter in color than the first tube, is because we're serially diluting the solution. Each sample is more concentrated than the next sample, and the one on the right is the most dilute. Then we can take samples from the tubes and put them onto the plates, and this can be done through that pour plate method or spread plate method I mentioned earlier. Let's say I take these plates and incubate them overnight. The next day I pull them out and I look at the colonies, the number of colonies or colony forming units on the plates. Some of these are too many to count. This one is also very many. Remember, we said we usually count plates that have between 30 to 300 colonies, so maybe I'll count that one. It has 50 colonies. And then since I only put 0.1 mil of my sample on it, I would multiply by 10. And then my total dilution factor for that tube was 10,000, so I can multiply that by 10,000 to find my original concentration. That would be 5 million colony forming units per mil, and that's the concentration of my original sample. So after we perform the serial dilutions, we can perform the pour plate method or the spread plate method. Here we have the pour plate method, and what happens is we mix our sample with warm agar before it solidifies, and we pour that onto a sterile agar plate, swirl it to make sure it spreads out evenly, and then let it solidify. Once it solidifies, we incubate them in the incubator upside down for about a day or two, and you'll see colonies form both inside the agar and on the surface of the agar that can be countable after a day or two. In the spread plate method, you already have agar, sterile agar plates that are prepared. It's hard to tell in this picture. There should be agar on the petri dish, and you take your dilution from the serial dilution, put it on there. At first it looks like a big drop in the center of the plate, but then you take a sterile spreading rod and you're going to spread out your sample across the entire surface of the plate, incubate them upside down as usual, and after a day or two you'll have countable colonies on the surface of the agar. We also have another method for measuring growth called the most probable number method. Sometimes this is abbreviated the MPN method. This is used for very low density or dilute samples, such as drinking water or testing food. This is a statistical approach that's based on detecting metabolic changes like color changes in a series of broth tubes. Sometimes we're also looking at turbidity, cloudiness. Here's another look at that most probable number method. We're going to be performing this on our own water samples at the end of the semester. Here we have 15 tubes of some kind of metabolic broth. Let's say in this case they're looking at lactose with a pH indicator called phenol red. So the tubes start off as reddish in color when they're neutral pH, and it looks like they take pond water and they're adding 10 milliliters of the water sample into each of these 5 tubes, only 1 ml of the pond water into each of these tubes, and only 0.1 ml into each of these 5 tubes. They incubate at body temperature for 1 day, 24 hours, and then they see color changes and maybe even gas formation in many of these tubes. Then we compare these results with a table that is constructed using a probability model of the sampling procedure. And based on our reading the table, we'll be able to conclude how many bacteria we most likely have per 100 milliliter of the water sample. All right, our next checkpoint asks, what's a colony forming unit or CFU? That is a single viable cell or a cluster of cells that gives rise to a visible colony, and those cells should be identical to each other. What two methods are frequently used to estimate bacterial numbers in water samples? So we either use membrane filtration or the most probable number, or MPN method. We can also measure the cell density indirectly by measuring the turbidity or cloudiness of our samples, assuming that the cloudier the sample, the more light will be absorbed using a spectrophotometer. And we'll be doing this in lab as well. In dry weight, we take a sample of cells and we're going to filter them out, spin them down, and wash and dry them before we take the measurements. We can also use a few newer indirect methods, such as measuring ATP production, the synthesis of proteins or nucleic acids, or even the rate of oxygen consumption to estimate live cell activity. So in spectrophotometry, using a spectrophotometer like the one shown here, we can use turbidity or cloudiness to estimate the cell density, as long as we have enough cells to produce turbidity. In the spectrophotometer, we have a light source, a lamp, a prism that allows us to select a specific wavelength or color of light that we want to be shown through the sample, and a detector to show or to detect how much light was able to pass through the sample. Theoretically, a clear sample with no cells will allow most of the light to be transmitted, and that's why this pink bar is thicker. A tube with more cells, that is more cloudy or turbid, will allow the cells to absorb that light. So less of the light will be passed through or transmitted through the sample, and that can be detected here. The greater the turbidity, the greater the number of cells. And we can use known samples and known densities to generate a standard curve to compare our unknown samples by. So what is the purpose of generating a calibration curve when estimating cell count from turbidity measurements? That's through our spectrophotometer. This allows the absorbance or turbidity to be calculated with actual cell counts, known cell counts. And that can be compared to our unknown sample to allow us to estimate the cell density we have. In the second one, some newer methods or newer indirect methods of counting live cells include measuring ATP production, protein or nucleic acid synthesis, or oxygen consumption. Besides binary fission, we also have some alternative division methods. There are several, and I'm only going to show you two here. The first is budding. Budding is shown here at the bottom right. This is when a small new cell grows off of the parent cell and detaches later. So unlike binary fission, these are not the same size. So you can see this is the smaller new cell. In fragmentation, shown on the left, filaments break into multiple cells that will grow independently. Looking at the difference between fragmentation and budding, in fragmentation, parent filaments split into multiple cells at once, and this can happen in microbes like cyanobacteria. In budding, a small new cell buds or grows off of the parent cell and detaches later, and we often see this in yeast cells. And that takes us to the end of our first video for Chapter 9. In the next video, we'll finish off 9.1 with discussion of biofilms.